All right, so I've got 8 o'clock. I'm gonna go ahead and kick us off if that's all right. United Poodle Association Education Committee had an opportunity to get Barbara to be a guest speaker, and we asked the members to narrow down the topics that we would like to hear about. And so the new color test came out as a clear #1 request for an educational opportunity here, and we want to welcome everyone and thank Barbara for taking time to put together this presentation and spend the evening with us.
Okay, well, thank you so much for inviting me to do this. And I'm going to share my screen. And I have a PowerPoint presentation that, let's see, PowerPoint slideshow. That I'll use to sort of talk about these different new color tests that have been developed in the last few years. There are 3 basic ones I'm going to talk about, and I'll stop after each section and ask if people have questions. You also are welcome to ask questions throughout the talk, and then it's about 30 minutes, so there'll be plenty of time at the end to ask questions as well. Okay, so what I'm going to talk about today is the why, how, and where for some new color tests that are the result of research that's been done just in the last few years and recently published. And so you may not be aware of all of them. So why do we care about color tests? So color doesn't impact the health of our poodles, so that's not why we care. But you may be trying to optimize in your breeding program either particular solid colors or particular multicolor patterns. And without a DNA test, trying to plan breedings is trial and error. Okay, so basically guessing what you're doing based on pedigrees. And with the DNA test, you can more easily move toward any color goals that you actually have. So there have been tests commonly available for decades now, and I'm not going to talk about general color genetics because that's something that people felt they were pretty comfortable about. But if you do have any personal questions about just general color testing or general color genetics, I'm more than happy to talk to people at any time. But just to generally review the kinds of tests that you may have been using in your breeding program, there's a test for B, which tells whether or not a dog can produce brown or is brown. E, which tells whether or not a dog can produce shades of red or cream, and that would be red, apricot, cream, white, anything along that line. An EE dog, small EE dog is actually some shade along that continuum. The K gene, which tells whether or not a dog can produce patterns at A or not. And then A, of course, tells what type of pattern a dog that has a particular version of the K gene will show. So this is where phantoms, sables, agouti, that's the A gene, determined by the A gene. And then, of course, whoops, why is it not letting me do my last one? There's the S gene, which will tell whether or not a dog that can produce party. And these tests have been available for years, and there's a number of different labs that offer these tests, and I'm sure a lot of you have been using them in your breeding program. But there are some new color tests. There actually are 3 new sets of tests that are available, and that's what we're going to discuss tonight. The first one are tests for silver and some blues. The second are for new patterns of agouti, especially creeping tan, for those of you that breed phantoms. And then genes that affect the intensity of red. So again, we're talking about the continuity from white to red in poodles. And this is also true for the tan color on any kind of phantom or sable. And for each of these things, I'm going to talk about briefly how the new genetic variants were found and what the new tests can tell you as a breeder. And then finally, where they're currently offered. And I see we do have a global population, and I'll tell you right up front, right now, the only tests the countries that offer these tests are the United States, but they're very new. And so hopefully they'll be available worldwide, not too long from now. Okay, so we're going to start with silver or progressive graying. And of course, this is my favorite gene because I specialize in silvers, or particularly like silvers. And everyone will still tell you on Facebook or elsewhere that there is no test for silver and this gene is not isolated. It's not true. Okay, so the mutation has been identified. It's been a couple years now, but the test is complicated. Okay, so there isn't really a direct test. And so this is sort of partially correct, but we do have some tests that we can use. So why is this the case? Why is it that the test is complicated? Well, let's start out talking about how it was discovered. And it actually wasn't discovered by looking at silver poodles. They were actually looking at shades of red in Nova Scotia duck-toling retrievers. So Danica Banish at the University of Davis, UC Davis, whoops, let me find my little thing, UC Davis Vet School, actually is a toller breeder. And her lab did a whole genome association study, which I'll show you examples of later, to determine what might differentiate different shades of red. So tollers can come in light colors, intermediate colors, or dark colors. So when they did this, the strongest association they found was variation at a gene called KIT ligand, or KITLG. And when the lab analyzed the DNA from dogs of different intensity, they found not just a mutation, just a little change in the DNA, which is what most people find, but instead there was a chunk of DNA that had different numbers of copies near the KIT ligand gene. And instead of affecting color intensity, let's see, I'm going to try to get rid of this, hide this, nope. Instead of affecting color intensity directly, what it did was it affected the gradient of color along the hair. So what's shown here is a figure from their paper published in 2020. And this is pheomelanin is a red pigment found in dogs, and eumelanin is black or brown pigment found in dogs. And what's showing here is 3 tollers with increasing numbers of copies of this sequence near KIT ligand. And what you see is actually the tip of the hair, the color is the same color red, but what's different is these dogs with lots of numbers of these copies have red all along the hair, whereas these fade towards the roots and these fade even more toward the root. And we saw that also with black or brown pigment, in that the one over here at the far right is actually from a flat coat retriever, and it's black all the way from the tip to the root, but the border collie is actually lighter at the root. So we can understand this actually interestingly. But what you want to know is, who cares about tollers? What does this have to do with poodles? And the Banish lab actually realized that for poodles, this would behave differently. Since KIT ligand affects the hair as it grows, they hypothesized that it explained fading of color in poodles like silver and silver beige. And that's because their hair grows continuously. And as the hair would grow out, it'd be getting fainter and fainter in color, just like it would in the sort of normal dog. In a normal dog, the hair grows out, starts out dark, gets lighter and lighter and lighter, but then it stops growing and it falls off, it starts over again. Well, the poodles, it just keeps growing forever. And that's what they found. When they took poodles that were black, they called these silver. I suspect they had some blues in with their silvers, cream versus red. And then what we're looking at here is the number of copies of these sequence. And what they found are blacks and reds, the really intense colors, had high copy numbers of the sequence, and the silvers, and to some extent the creams, had low numbers of copies, almost like 1 copy per chromosome there as well. So it's actually pretty interesting from a scientific effect, in that we can understand why this particular gene has this effect. KIT ligand is called stem cell factors. So you've probably heard of stem cells, and they're cells that actually move in and will take over when cells have been damaged and produce new cells. And it's actually known to help cells that make pigment to actually survive. So if you have a lot of KIT ligand, then your pigment cells are going to live longer. And so as the pigment cell is the cell that has pigment cells associated with it is making hair, it's going to have pigment cells there the whole time. So the hair is going to be dark along the whole thing. Whereas with less KIT ligand, so lower numbers of copies of this sequence, you're actually going to stop making dark color. And actually, this explains this unusual aspect of silvers, is if you damage them, then their skin, the new hair will grow back dark, the burnt black or brown if they're silver beige, and then it'll gradually will turn to silver as it grows out. This is my silver beige where I've actually clipper burned her, not on purpose, but accidentally. And then this area will be dark brown until it will grow out over a period of a couple months, and then we'll turn back to this actual color here, because these new cells come in with new cells that have a lot of KIT ligand, and it's only as that amount of KIT ligand goes down that they actually start to turn gray. So why don't we have a silver test? If there is a gene and we know what the mutation is, why isn't there a silver test? And the problem is that most DNA testing looks for specific sequences in DNA, so specific changes in DNA. You have this little base called C here, and instead now it's an A, and that's a mutation. But in this case, we don't have that. We have different numbers of copies of the same sequence. And the technology that's used for DNA testing can't detect that. And actually, the technology to actually look at numbers of copies is difficult. And so that's why there isn't currently a good mutation test for this. Now, according to Dr. Bonish, who's the lab that discovered this, VGL at UC Davis is working on a test that they offer. They've been doing this for a couple of years. But right now, we can still get information from a linkage test. Embark offers a linkage test, and they include that in their intensity determination as well. So what's a linkage test? Right now, this is the only thing we have available if we want to actually look at our DNA test for silver. But the problem is that a linkage test only looks at DNA sequences near the mutation, but not the mutation itself. So what it relies on the fact, now, some of you, I don't know if any of you are old enough to remember when we didn't actually have a PRA test and all we had was a linkage test. And it's not always right. It can be inaccurate. Okay, there's a higher than average mistakes, but it was better than having no test at all to know whether or not your dog possibly carried the PRA mutation. But when in a linkage test, you don't know what the exact mutation is, but you find DNA sequence differences that are close to it, that are linked to it, that are inherited with it. That's all that actually means. So that the affected dog would have an A and a G at these positions, whereas unaffected dogs have T's and C's at this position. And so we would look at the A and say, oh, this dog must have the mutation. And if we saw a T, we'd say it doesn't have the mutation. But because we're not looking directly at the mutation, sometimes we're wrong, but a lot of the time that we are. So although this isn't perfect, right now this is what we have for silver. These, unfortunately, Embark is the only lab at which you can get this test. Before I go on to the next section, are there any questions about this particular test or mutation? I'll open the chat box in case someone has something written.
I will ask a question.
Okay.
So I've been using Embark for a while, and I know that there have been some issues and some questions, and people have said Embark's not reliable. The one that I had was with Brindle, so my boy's KB, and then I tested it with I think BetGen, and he's actually KYKBR.
So Brindle is the same kind of problem as silver in that it actually isn't a DNA sequence difference. It's the number of copies. And it's even worse than silver in that it depends where the particular copies are on which chromosome. And there actually is no way that anyone can determine that. So brindle is caused by having one version of K, KB, and one version of K, KY, on the same chromosome next to one another. So the problem is that they have both a KB and a KY, and you can't tell whether there's a KB on one chromosome and a KY on the other chromosome, or a KB and KY on one chromosome and a KB on the other chromosome, or KB, KY on one chromosome and a KY on the other chromosome. You can't distinguish them because it's different numbers of copies. So it's a technological problem. There's one lab now in, I think, Hungary that is actually found a way to count copies. And they're actually offering a brindle test, but they're the only ones I know of that actually have been able to do it. I actually realized, I think I have one more. Oh, let me go through this and then I'll come to the question. So there actually are further complications. So I was wrong, that was the end of that. One is that this KIT ligand mutation is probably not the only gene that predicts fading. It seems to predict silver, but there are actually blues that fade differently and can't produce silver, and they don't have this KIT ligand, the same KIT ligand sequence as silver. So we need to know what that other gene is. Okay, so more science is needed for that. If fading in brown might be KIT ligand, or it might be this strange blue thing. Okay, and we don't know that. We need more tests done. And despite this, I actually have been able to successfully use this in my program to tell which puppies are going to be blue versus black versus silver at birth. Or where I find it most useful is here's an EMBARK results for a puppy looking at KIT ligand. They reported under red intensity, okay, because that's where it was originally discovered, but it does cause silver or silver beige. And to know whether or not white puppies can actually produce silver or not. So how is intensity involved with, let's see, I know at one point you're, okay. Yes. So so lots of people don't like EMBARK, and there are some things I don't like about them. But one thing they do is they might introduce a linkage test too soon, so that's not very accurate, but they continually improve their model the more dogs that they test. So early on, it didn't work very well, especially for standards. Now it seems to work better. Okay, so all we could find for blue with Tasha you and a bunch of other people had submitted some EMBARK data sets. All we could find was KIT ligand. I think there's another blue, but I think what's going to have to be done is a study where we have those blues that can't produce silver and compare them directly to blues that can produce silver, and then pick out what that actually is. So the old tests for brown and cream are fine for turning brown and cream. It's just that, for instance, the cream test can tell you the dog's going to be some shade of cream, but they can't tell you what shade it's going to be, whether we're talking ice white or we're talking red. And we'll see, I'm going to come back to the red intensity test on that. Okay, I'm not sure I understand your question, Suzette. So maybe we can come back to that at the end. All right, let's go to the next one. Okay, the next one's a Goody. So if you breed solid color poodles, you're not interested in the Goody. But if you're interested in multicolors, then this is what determines sables and Goody or phantoms. And we thought that a Goody came in 4 versions. One of them, AW, was the normal wolf version. And then there were mutations, AY, which is sable, AT, which is phantom or black and tan, and A is something called recessive black. But the Goody locus turned out to be a lot more complicated than we thought. Now, we sort of knew this because there were there's an observation that some sables are shaded with black and some are not, but both of them come up to be AY. Okay, so there's something that has to explain that. And some black and tans are mostly black and a little tan, or some have what's called creeping tan, or say more tan on their body, and both of them just show the AT mutation. So we haven't known exactly what explains these differences. Now, a paper in 2013 published said that the second, so in other words, tan versus black and tan versus creeping tan, was due to another gene called RALI, but there's nothing to explain shaded sables. Okay, and all that changed a few years ago in 2021 when the Banish lab again actually looked at agouti. Now, don't worry about this picture, okay? It's kind of here for eye candy, all right? So I'm not going to go through it, but I wanted to put a picture in here. But what they did was they actually went and took skin samples from dogs of different patterns and different parts of their body and looked at the production of the A gene, which is called ACIP. And what they found was there were differences in the exact kind of ACIP that was made and how much of it was made depending on the dog's patterns. Then when they went in and sequenced the DNA of these dogs of different patterns, they found these sequences nor signs in different patterns. So these are signs, okay? So some dogs would have a sign here and some wouldn't. And then some would have these multiple signs, all right? And they were actually associated with different patterns that were found in where you found actually tan and where you found black. And these different signs were determining how much of that ACIP gene product the skin was making, and also exactly what type you were making. So sometimes you had this part of the gene and the rest of the gene. Sometimes you had this part of the gene and the rest of the gene. We actually were getting several different gene products out of that individual gene. Okay, so what we knew then was that there were 4 types of agouti locus, and now we know there actually are more. So the agouti and recessive black, they haven't changed. Okay, we know there's one thing that actually determines those 2 different phenotypes or the ways that dogs look. But there actually are different DNA sequences that determine whether you're going to be fawn but no black shading versus sable with shading. And then there are going to be different DNA sequences that say you're going to be black back is what they call this, which is what we would call phantom, or black saddle, which is what we would call creeping tan. And now you can actually determine what your dog has for all these different possibilities. Now, right now, VGL at UC Davis is the one doing the testing, but all of these have been published, and so other groups could start offering this test as now. Now, what about Raleigh? So Raleigh supposedly was creeping tan, but it was probably a mistaken identity case, okay, which happens a lot of times in science. So Raleigh was found associated with creeping tan in basset hounds and corgis, but didn't seem to work as a predictor, particularly in poodles. But if you look at where the gene is, it's actually really close to ACIP. All right, so I'm going to show you, get you some more science here. So this is actually a piece of the dog genome at a certain focus. And this is supposed to represent the entire chromosome 24, this big gray bar here, which is where the ACIP gene is found. And this little red box here is now blown up over here. And so it's a very small section of the chromosome. And here is the Raleigh gene. And right next door to it is the ACIP gene. Okay, so remember that whole linkage thing? Probably what happened was that this mutation in Raleigh was actually just a meaningless mutation that was linked to that particular combination of signs that's found at ACIP that makes creeping tan. So at this point, you can ignore Raleigh, but if you care about tan versus creeping tan or different forms of sable, you can now get your dog tested for these through UC Davis. Okay, now we're on to our second one, red intensity. So are there any questions about the second section? I thought that one was probably of limited interest, but for those of you that are trying to optimize particular phantom patterns, it's definitely important. Okay, so can I go on? All right, so red intensity is of interest to people often, whether you're breeding solids or multicolors. And we know a lot now more than we used to. But the bottom line shows that it's not completely clear. The bottom line is that variation in anywhere from 1 to 4 genes determines the intensity of white, cream, apricot, and red in poodles. And as I said, this is true for the intensity of tan points and sables, as well as solid dogs that are just white or red. Now, a lot of people have tested for what is supposedly intensity gene, and this is a gene called MFSD12, and a variety of labs, paw print labs in Europe, VGL, just about a lot, Gensol. Most labs will actually give you a test for this, but it turns out it's probably not the most important gene for poodles. It may have only a minimal effect on intensity. The reason that I'm saying somewhere from 1 to 4 genes is that different experiments currently have concluded there are different numbers of genes involved. So this is a situation where we're not really sure, and we need to do more science. Okay, so the first analysis I'll talk about, even though it was published, actually it was published before the first one, found that there were 5 genes that appeared to affect red intensity. So this paper was published in 2021 from the Boyko lab, who is associated with Embark. What they did was they analyzed over 3,000 dogs of different breeds by GWAS to try to see what intent they had photos to work with to assign an intensity of red to color to the dogs. And what they found were there were 5 genes that they found association to. So one of them was KIT ligand, and we already know about that because we saw that with the tollers, and that does affect red intensity. This other gene, I, okay, which has previously found to be important for red versus white in white shepherds, poodles, and Bichon. So that was a known gene. But then 3 new locations, one on chromosome 2, one on chromosome 18, and one on chromosome 21. They didn't actually determine what the mutations were, but they did actually localize where on the chromosome it might be. So if you do Embark testing of your dog and you look at the subloci for intensity, they'll give you reports on all of these genes, chromosome 2, KIT ligand, chromosome 18, MSD12, and chromosome 21. Now this dog here is actually a very, very light cream. She's almost white. Okay, and you notice that actually most of her versions are what are found in cream dogs versus red dogs. So one question asked though is, are all these relevant to poodles? Okay, because the study was done in all breeds, and it might be that all those variants are not found in poodles. And what the Slavny et al. Did was they looked to see if variation was found in different breeds. Okay, so don't worry about this. We're going to look at what's important about it. And they actually looked at a variety of different breeds, and they go from breeds that are white, so American Eskimos, Samoyeds, Bichons, to breeds that are red. So down here we have Irish setters, the reddest of the red, and the red-bone coonhound. And then look to see how much variation there was for particular mutations that they'd found in these 5 genes. So the poodle is here. And the poodle, they said, was intermediate in intensity, meaning that they had some whites, they had some creams, they had some apricots, they had some reds. And if you looked at that whole group of, let's see, 53 poodles, there actually was an extensive amount of variation found in all of these particular genes. Now, the color coding is, if you have yellow, that means that all of the dogs have the mutation. So for instance, Irish setters, all of them have a mutation at chromosome 2. All of them have the ones at 15, all of them have the ones at 20, and all of them, some fraction of them have it at 22, but none of them have the mutation at chromosome 18. Poodles actually have all of them. And not only that, if you look at chromosome 18, well, we already know what kit ligand does. Okay, so kit ligand's in here. That's chromosome 15. And we already know that's actually fading color after birth if your hair grows continuously like a poodle. But we also see actually chromosome 18, actually zero, blue means none of the dogs have the mutation. Okay, so most of these breeds actually don't have that mutation. Poodles are one, red bone coonhounds are another, and then I think there was, those are basically the ones that have it at any appreciable frequency. So it could be that all 4 of these genes, plus kit ligand, which we know actually leads to fading of color after birth, may actually impact color in poodles. Okay, but what happens when you do a poodle-only analysis? Okay, so the Banish lab did that. And this is a genome-wide association study. This is what the results actually look like. Okay, so I'm going to sort of explain what you see here. So what they do is they take a certain number of dogs. They had 79 standard poodles that were cream, white, cream, apricot, or red. And what they did was they looked at 200,000 different points of variation across all the chromosomes. And each one of these little points here is a statistical result for what kind of variation they saw for that particular piece of DNA. Okay, now there are 200,000 little points here. You don't see them, okay, because most of them are close to 0, meaning that they actually don't differ between the dogs depending on their color specifically. But if you look here, these are chromosomes 1, 2, 3, 4, and then they're put in here where they are, you know, across that span of the chromosome. If we look over here, woo, there's a place here where there's a lot of variation found between dogs based on color, and lo and behold, it's on chromosome 18. So the Banish lab did their G-SWAS on 79 standard poodles. And you might be upset about that, but they found this one associated variant on chromosome 18. They repeated it with miniatures and toys, and they got the same result. And found that there actually was a mutation they could find on chromosome 18. It's something called a retrogene, which is a gene from somewhere else that got copied and put into a new place on chromosome 18. And so when you do that, that can actually lead to a different expression of the gene and then have it have a different effect that it would in its normal place. So clearly chromosome 18 is most important in poodles. And what they actually conclude is that it's the only gene that's important in determining color of red in poodles. Well, which one's right? So Embark says 4 plus kit ligand. And that was looking at all breeds that do vary in poodles. And then the Banish lab says 1. Well, it's kind of hard to believe that it would be only 1. Chromosome 18 is clearly the most important for birth color. But in my opinion, anyway, it has to be more than 1. Because if it was only 1, then dogs could have 0 copies, 1 copy, or 2 copies of the mutation. And you would expect there to be sort of 3 types of color, and we don't. We have this whole range of variation. Okay, so it's likely that some of these other genes actually impact color as well. So 4 might be relevant, may not be relevant, but they do vary in poodles, and I think it's unlikely that chromosome 18 is the only important gene. I bet, based on some results that we've done, is that chromosome 2 and chromosome 18 and chromosome 20 are important, but chromosome 21 may not be. So this is, I couldn't find my more recent results. This is just an example of some dogs that have been tested in Embark. Okay, and they're just sorted by their color. So here's 4 reds, 2 apricots, 2 light apricots, sort of apricot-y creams, and then some whites at the bottom. And then this is what Embark said for chromosome 2, for kit ligand, for chromosome 18, for MSSD12, for chromosome 21. And if you look at the reds, you see a lot of red here and very little cream. Okay, and we actually expect them to be red at kit ligand, meaning will not fade, because if they end up red as an adults, they're not fading, right? As we look at the apricots, we start to see more cream showing up for all of these. at least all of these. And the same thing, we get a little bit more creams here. And then the whites actually are cream only for these 3 genes. This, remember, is kit ligand. It's going to affect whether or not they fade. And we know some whites can carry silver and whites cannot. So this doesn't bother us. And this is why I think that chromosome 21 maybe isn't relevant, in that we'll often find dogs that seem to be red that are actually white for chromosome 21. But it should be possible to collect a lot more information and then decide which of these actually are more important. So I'm just going to go back to that slide one more time. So if you do embark your dogs and you do care about intensity of color, start looking at this, because definitely chromosome 2, chromosome 18, MSDS12, they're going to tell you whether your dog is actually black or brown or red or, white, red, cream. It's going to tell you what they're capable of actually passing on. Okay, so I've said a lot of things, and hopefully some of it made sense and some of it was interesting. So what is there to take away from this? So one thing is that coat color science, like all science, is not a bunch of facts, but it's changing, expanding information with time. Okay, so it's important to keep up with things, to see if there are things that might be useful, or see if things that you're doing that aren't right. We can test for silver now. We can test for creeping tan and different shades of red now. Some of the tests are not perfect, some of them are, but we at least can get information at this point. If you want to breed your red to blacks to improve your head, to improve your coat, you can do this without worrying about color if you can find blacks that actually have red intensity genes present, and you can test them now. So you could take 2 blacks that you're considering, embark test them, look at their red intensity genes, and pick the one that best complements what you're trying to do. Similarly, as a silver breeder, if you want to breed your silver to white, you might get all blues because some whites don't carry that kit ligand silver variant. But now you can look at your embark test and often see whether or not you'll be able to get silver or blues. And with the toys that I've done, it's been almost 100%. There's one dog, a blue, that is supposedly a black, but she can't produce silver. She's been bred several times to silver and doesn't produce it. So she's one of these sort of different kinds of blue. In the case of silver and shades of red, unfortunately, embark is the current test to use. Just because shades of silver, we don't have a direct mutation test yet, though VGL says they're going to be developing one. And for shades of red, there's no reason why other companies couldn't test for these different genes. They have not started to do so yet. And for creeping tan or different types of sable, VGL is the current place where you can get a test. As I said, VGL will probably expand their testing, and so they would be an option. And I just want everyone to know I have no association with any testing companies. So the reason that I'm focusing on these 2 is only because the research that uncovered these mutations actually were associated with these companies. And so they're the first to actually offer the tests. So thank you very much for your attention. I'll get off here. I'm happy to, I'll turn off the PowerPoint and I'll get off. I'm happy to answer questions. I got one in the chat, looks like. I'm also happy to answer questions by email or by DM through Facebook. I'm always happy to talk about genetics. So if there's anything that you want to ask that you don't think is sort of generally interest to everyone, please don't feel, awkward reaching out to me. So let's see, I'll stop sharing there and see. Oh, how can we find out more about it? I would donate DNA samples from our poodles for their research. Good question. So actually, she often comes to PCA, and I don't know right now, a lot of the work that she's doing with poodles actually is on the CDDY IVDD mutation. So that health mutation that affects intervertebral disc disease. And there's actually some interest on her part on an MRI study using poodles. So I'm sort of been talking to her about that. But if there's anything that you think is worth looking into, then let me know and I'll be sure to pass that along. Right now, her lab's doing most of the color stuff, that or Embark. And in the case of Embark, again, if they can get interested in it, they have all the data. They just need the phenotypes to actually do that. Oh, okay. So can they explain more why apricot red lose the color with aging? So it's possibly one of three things. Okay, so one is it could be due to KIT ligand, that variant that is actually causes silver. Okay, and so what happens is as the hairs continue to grow as the poodle ages, then there are fewer and fewer melanocytes there to actually put the color in the hair. So the color just fades as they go. So they'll start off with a particular color and it will fade. So it could be that. And again, now we can sort of ask the question of whether or not that is true for a particular apricot by testing for KIT ligand to see what's there. Second one might be that other thing that fades and isn't actually silver, okay, which we don't know. People who breed browns are pretty sure there's something separate from silver that fades brown. And it could be that it's that's affecting the apricots, or it could be a third thing. Okay, and until we actually get dogs tested for things that we know about, then we don't know when we don't know, right? So it'll take some time. Oh, the rufous gene. So I actually had some students try to look for the rufous gene this past spring, and we got some. We had a bunch of Embark data on dogs that had gotten darker or had not gotten darker, and then we also looked at them phenotypically to see what genes actually seem to be important in determining color. So as far as what genes that seem to be important for determining color, we found chromosome 2, chromosome 18, and then in a sort of case-by-case basis, it looked like chromosome MFSD12 was also important. Okay, so we sort of got support for those three for Embark. But we could not find anything that was different about the ones that got darker versus the ones that got lighter. So my hypothesis was maybe that it was like that chromosome 21, and that's why it behaves differently, but that did not show an association. But unfortunately, we had a total of about 60 dogs, and about 12 of them were ones that got darker, and I don't think that was a big enough sample size. So again, that's something that if you can get Embark interested in it, their worst problem is knowing the phenotype of dogs. So there's a survey you take about your dog, and they have to be, not everybody does it. Okay, so then they don't have information on what your color of your dog is. And people don't necessarily know what color their dog was, right, at birth, and then it got darker. So I keep trying to convince them they need to work more with breeders who actually know what the colors are, as opposed to just, these 3,000 dogs they have, and they have a picture, from some pet owner as well. So we don't know. Oh, I still darkening at 6 months. Yeah, I didn't believe in the rufous gene until I had a litter last year where I looked at the dog when it's born and I said, this looks like what people say the darkening ones have at birth. And sure enough, he got darker. And of course he'll fade because he has silver. But yeah, there's definitely something there, and we don't know what it is yet. So anything else? Oh, high intensity. Okay, so I really hate it when people use the numbers. So for red intensity, they say, oh, my dog's at 8 out of 10. My dog's at 10 out of 10. My dog's at 4 out of 10 for the red intensity numbers. It's not the way things work. Okay, because for one thing, we just saw KIT ligand doesn't really affect color for poodles. It's fading of color, right? So you could have 8 out of 10 or 2 out of 10 and they be at KIT ligand and be white because everything else is cream, or you could be 2 out of 10 at one of the others and you've got some red to you. Okay, it depends. It depends which gene you actually have the mutation, and they're not equivalent to one another. Same thing with breeding. You could have 2 dogs that are 5 out of 10. They could end up with all 5 out of 10 puppies. They could end up with puppies that actually have, more than that, because it depends on where the mutations actually are. So a 10 out of 10 says it's red at everything. It should be red and it won't fade. That's pretty much what you'd say. But anything other than 10 out of 10, it's not really meaningless. You have to look at the individual genes and then decide how individually they're actually going to contribute in the new one. So in other words, chromosome 2 from one parent, chromosome 2 from the other parent, only one of them is going to go from this parent, one of them is going to go from that parent. It's so that they don't all assort all together, all 5 together. So let's see, is there anything else? Any other questions, verbal or? Was that helpful or useful?
Yeah, I think that's great. Having apricots and blues, I had a lot of interest in this, and I'll be looking at those and BERT tests and seeing what I can figure out.
Yeah, I'm hoping that these tests will help people feel a little better about mixing colors, but interesting things happen when you mix apricots and blues sometimes. So
definitely not mixing those two.
Well, I've got a little bit of everything in mind, I must admit. I had some red that came in from one line, and then I try to have silvers, but sometimes my best dogs are blues, sometimes their best dogs are creams. I'm not going to throw an incised toy out because they're not my favorite color, that's for sure. What about brindles that fade? Okay, so I don't know about the losing their stripes. It's other than if they have kit ligand, cream, or even red cream, which would be sort of the blue equivalent, they're gonna fade. So it's whatever the black brownish color is gonna fade, and whatever the tan color is gonna fade. So it's probably just kit ligand just operating there. Black intensity, yeah. So that's a real interesting question about the inky black versus bad black too. Another interesting, we don't know enough about what those red intensity genes actually do as sort of to create the differences in intensity. So it's possible some of them might affect eumelanin as well, so which is black or brown, but we don't know enough.
I think it's interesting too, seeing, I mentioned that my boy ended up actually KY, bred to a brindle party. He produced black brindles. So instead of getting lighter, she got darker.
Yep. And we don't know what that is.
Yeah.
Was his black getting darker or just his tannish region?
So the puppy was born with tan and black stripes, and has by about a year old, other than the long hair, the hair at the root had all gone black.
Yeah. The other thing we don't know is what determines the variation seen in brindle. Brindle is really interesting in that it's actually probably epigenetics. So what it is when you have 2 genes next to one another or copied, repeated, lots of times that actually is a signal to the cell that there's something wrong, and it'll shut off one of the copies, thinking it should have only one, so it's going to shut one off. And so with brindle, sometimes it shuts the KB off, leaving the KY, and then you'll have a sable. And sometimes it'll shut the KY off, and you'll have a KB, and you'll have a black. So we're not exactly sure that's how it works, but that's how the people think it's work. And so why do we have these reverse brindle, where it's mostly black and a little bit of tan, versus mostly tan and a little bit of black? That's not not known. It's a really interesting question scientifically.
I actually had a friend who raises Caucasian shepherds, didn't know it and had a litter of brindles. And so she was asking me about it.
Yeah. So one question is, white better to breed with apricot red than black for not to lose color intensity, or does it not matter? So are you talking about intensity of the black pigment on the nose and everything? So if you're going to breed a white with an apricot red, you're only going to get something in the white apricot red range. And it's going to depend how those 4 genes, actually end up giving creams versus reds to each one of the pups, right? So you're going to get some cream, apricot, something along those range. When you breed to black, well, you don't know what you're going to get. If the black can actually produce whites, okay, which would be a big E, little e dog, so the E gene there, you won't know unless you know what those color intensities genes are. Now, black pigment seems to be somewhat separate from something, good pigment versus bad pigment. And I've seen that with my silvers, because some of the silvers will have good pigment and will produce this wonderful pigment in whites. And then some will have what seems to be perfectly fine pigment and produce poorer pigment in whites. So, and that's even with like silver by silver crosses. So there's something else that also affects nose and eye rim pigment that we're not completely sure of. Well, if there aren't any other questions, I guess we can close up shop, or if we can hang out for another couple minutes.
Yeah, it looks like things are quieting down, and from the United Poodle Association and the Education Committee, we thank you very much for your time and preparing the presentation. I myself think this was absolutely fantastic. Thank you.
Well, thank you everyone for coming. I really enjoy it.
Thanks, Barbara. We appreciate you.
Very good information. Thank you so much.
Thank you for this.
So Angelina, you asked about apricot and brown. That some people think that that's a way to intensify color, but I don't think there's any science basis to that. It may be that those browns actually didn't have fading, and that's why it was actually helping to intensify color. The problem with that, of course, is that you can end up getting brown noses in your offspring, which is not preferable. I don't know if it's allowed in FCI. It's allowed in AKC, but it's not preferable. So. I wonder if Embark and VGL are all of a sudden going to have an increase in test kits.
Yeah.
I should have started with the caveat is I don't have any connections for any testing companies. How would you rewatch this? I believe it's being recorded.
Yes.
And so, Michelle, how are you going to make the recording available?
So the recording will be posted on the United Poodle Association website under the Education tab, and it will also be shared on Facebook. I don't know if it'll be a link to the website, but then that will be shareable so people can share that with others. All right, I'm going to go ahead and turn off the recording.